We've previously spoken about how you can perform cloning using PCR. And in order to do that, you need to create primers or design primers that allow this process to happen. This contrasts the traditional approach, which is to use restriction enzymes. For this workflow, two types of primers are typically used. So you want a detection primer. This primer is used to test for the presence of a gene because it's designed to target regions in your gene of interest. So it will look for the presence of your gene in a plasmid, for instance. But then you would also want to design the actual cloning primers. So the first step is to get the sequence data for the region you wish to amplify. You need to include up to 10,000 base pairs, so 10 kb, of the genetic context that the region is in. So the sequence can be a plasmid or a segment of chromosomal DNA. And you want to go to Primer 3 Plus for this cloning primer design. In Primer 3, you want to click the tab to go to the General Settings tab. Here you change the default product size for your detection primer to be about 450 to 550 base pairs. This should be sufficient for detecting your product. This field must be a range. You can't just put a single size. But if you really want a narrow size, you can still say something like, say we want 89 base pairs, you could just say 88 to 89. For the primer melting temperature, I've always been told that you allow the polymerase that you're about to use to determine what the primer melting temperature is. However, it's a great idea to aim for it to be around 60 degrees Celsius, according to Sebastian at Tiny Green Cell, who's giving these instructions for designing the detection primer and the cloning primer. So click back into the main tab, and here you want to copy and paste the entire sequence that you want to amplify. So if it's from the plasmid, the entire plasmid you wish to amplify. If the plasmid is higher than 10 kb, so 10,000 base pairs, you want to select a 10 kb chunk where your gene of interest is in the middle of the chunk. You want to include as much genetic context for your primer because you want to avoid the primer binding to other regions you're not interested in. So you're going to get additional products that you're not interested in. If you only put in your gene of interest, you have a higher chance of the design primer binding to other regions of the chromosome or even genome. You can take a look at the sequences of your plasmid via a plasmid viewing software. Softwares like Benchling or SnapGene allow you to view your plasmid. You can copy your gene of interest and place it into these softwares. So once you've looked at your full plasmid sequence, you can now copy your gene of interest. And we're going to now place that gene of interest into Primer 3 Plus on the main page. So the plasmid viewer will allow you to copy the gene of interest. And on the Primer 3 Plus page, we've already got the gene of interest plus the genomic context up to 10 kilobases, 10,000 base pairs. So here we're trying to verify that the plasmid sequence of interest will be on the gene sequence that we've pasted into Primer 3 Plus. So the copied sequences, now we're going to try and use a search tool to be able to see if it exists in the gene of interest and the larger 10 kb context that we've given it and placed in Primer 3 Plus. So use the search function in Primer 3 Plus on the main page to find the sequence that you have copied from the plasmid. So this should result in the sequence being highlighted in Primer 3 the primer 3 plus in the text box. Now you want to drag your cursor over the highlighted region to highlight it again. And now you want to click the included button. So this included button is something that results in numbers being generated in the included region text box, which is just below where you've pasted the full sequences in primer 3 plus. So what it's doing is it marks the start and stop position of the region to include in the primer search. So this is essential because by using the included command, you flag to the software that it must include the bracketed sequence. The software looks for ideal primer sequences inside that region. Okay, it will also take the outside sequences into consideration, and you want that because this allows it to avoid off targets. So now, if we've done everything correctly, we have our parameters. We've told it, the size of the primer that we're interested in. We have a template sequence which it needs to design against, and we have the region that we wish to design the primers for. So the only thing left to do is to request that the software picks the primer. So click on the pick primers to initiate this. So we should have some results now. The best scoring primers, you'll be given 2 options at the top, and these are the best given the context and the thermodynamic considerations. So copy the recommended primers, but remember to search for them in your plasmid just to make sure that they bind where they should on your plasmid. So use your search function again to make sure that the sequence of the primer suggested actually binds to the plasmid. Just a little bit of double checking, cross checking. What's cool to note is that the reverse primer is already in the reverse complement, so you do not need to reformat the sequence. The primers are also already in the 5' to 3' orientation, so this means that you can copy and paste the information straight into your oligo request Excel spreadsheet from your favorite oligo manufacturer. So that's it for detection primers. Now let's look at designing the cloning primers.
Be strong.